see some what protein folds mean and what are some of the topology in proteins. So protein structures topology is very much important for drug design. And when we are going to do more drug design aspects or go more advanced topics, you should have a fair enough understanding about how proteins fold and how protein folding works and what are the implications of protein folds in drug design. So there are a lot of things, but some of the topics in this or some of the slides in this particular talk in the first 1 hour will be a repeat of what has been taught in the previous sessions for you. But then, since Dr. Solanki said that you are from different background, so some of you may be very familiar with it, some of you are not, and that's perfectly fine. So we are going to look at the biophysical aspect of proteins, and that is what the protein folds and protein folding is all about. So some of the books that you can refer or whatever talk is there, the protein structure or protein folds in this particular talk, you can refer from White and White. It's a very good biochemistry book. Then there is an amazing book called Introduction to Protein Structure by Carl Brandon and John Toos. Excuse me. So this is a very good book. There has been no other book like this. There is no third edition that came out. The second edition, first edition are all like collectible items or collectible books, especially for some of the images. And I will show you those images. Okay. And then what we can do with protein structures, you have protein engineering books and protein design books. These are very basic books in the sense that they are written by eminent people who were involved in protein design right from like 40 years ago. So these four books, and then of course there are many other books as well, but these are very good reference books that you can start looking at protein structures, and especially for drug design and many other biophysical and mechanism of action and so on. So uh, rather than talking about proteins as molecules, proteins are can be thought about as a nano machine. They are nanometer in scale, and they are an efficient system that has been selected through many different trials in evolution, and they are very good at what they are doing in the machine through the biological function. So for a protein to function, you need a structure and you need the subsequent function associated with that structure. So there is a so-called dogma, which we will also see later on. But you can think about that this is a three-dimensional object, and this three-dimensional object is not something that happened by chance. There is a lot of evolutionary history behind hemoglobin or myoglobin or any protein that for that matter. Proteins do a lot many things. They do a huge diverse range of functions. They are involved in catalysis, which are basically enzymes. So anything that ends with an ase is an enzyme. That's the general rule of thumb. But of course, there is exception to the rule. So you have proteinase, you have keratinase. So keratinase are proteins that break down another protein called keratin. So these are very industrially important proteins. Most of the proteins as that work as enzymes are very important drug design purpose. They are basically many are drug targets. But then proteins do a lot more in terms of structure, involved in transport, involved in messaging, involved in immunity, involved in locomotion, involved in storage, and involved in receiving signals from outside the cell. So there are many, many different proteins. The protein universe, if we look at it, is much more vast than compared to the nucleic acid universe. And there are about 1 trillion proteins that we can think about. but we do not know all the 1 trillion structures, or we do not know all the 1 trillion sequences. So there is some amount of necessity for some computational methods that can fill up this gap. And there are many, many different ways in which we can do this with current AI-based methods. So protein synthesis is done with a protein called as a ribosome inside a ribosome, and this ribosome, this particular machinery of synthesis of proteins, is what got the Nobel Prize in 2009 by Venki Ramakrishnan, Thomas Seitz, and Ada Yonath for the ribosome structure and deciphering the mechanism behind it. So when we are looking at biology, we are looking at multiple skills. So multiple skills in the sense like we are looking at biology in terms of organ, in terms of tissues, in terms of cells, in terms of molecules and atoms. So there is a hierarchical way. And as you go down from left to right on the screen, the scale is more finer and finer and finer up to the atom level. So there are different ways in which we are looking at biology, and protein structures are at this level of molecules and atoms. It's pretty obvious. But to understand the communication between two molecules, you need to have the structure of the molecule. And that is why you need to know the arrangement of the atoms. And once you know the arrangement of atoms, you'll be able to understand how it works. And this is, as I was asking right before we started, the protein structure determination by either X-ray or NMR or cryo-EM, all these three different methods, and there is a free electron laser, the four different methods. They all are solving only one goal, that is determining the arrangement of atoms. So protein structure, if you take, this is typical biochemistry layout where you have four different types of structures, hierarchical. So you have primary structure, which is the sequence, which leads to a secondary structure, in this case, alpha helix. And then you have the helix, which you can see here, part of a much bigger entity, which is a tertiary structure. And in the case of hemoglobin, four such structures come together to form a quaternary structure. Not all proteins need to go to a quaternary structure, but there are many proteins that need a quaternary structure to function. So the basic unit of protein, again, all these are from talking the same things that has been covered in the previous talks, but I'm looking at in a different way. So the basic unit of protein are amino acids. There are 20 different amino acids that are frequently seen, but these are the L amino acids, the levorotatory, based on the chirality of the molecule, because you can see there is a chirality that can be established when you are looking down the carbon from the hydrogen. So this is the viewpoint, and you have the amine group and the carboxylic group present on either sides of the amino acid. The name itself suggests that there is an amine group and an acid group that gives the name amino acid. Now, there are about 300 different other amino acids that are synthesized, that are seen in nature. Some of them are not seen in nature, but these other amino acids are the D amino acids, unnatural amino acids. They are modified form of one of these 20 amino acids. So for our basic understanding about drug design, for drug design purpose, 20 amino acids, the ones that we talk about, the standard amino acids is more than enough to remember. But there is a lot of work that has happened and still happening on D amino acids and other peptides that can act as drugs and inhibit a lot of other pathogenic organisms. Because these are chemical molecules, you have the 1 letter code and a 3 letter code. The 1 letter code is shown in filled circles. The 3 letter code is shown below. And most of you may know this from basic biochemistry, that these 20 amino acids can be grouped into essential or non-essential, because essential are those that are need to be provided from the food source. They cannot be synthesized in our system. And the humans, non-essentials are the ones that even if it's not present in the external source, the system, our body can generate those amino acids. So we have evolved in a way to be dependent on many other amino acids that are shown in dotted lines. And being from looking from the biochemical aspect, because these are acids and with the amine group, you can classify them either as aliphatic, aromatic, acidic, basic, hydroxylic, or sulfur containing or amidic amino acids. So the color code tells us how many different groups are there. This type of grouping helps us in many things when you're talking about the protein structure. The reason is that one, just like in the nucleic acid, you can replace a purine with a purine or pyrimidine with a pyrimidine. You can replace one type of amino acid or a group of amino acid with the same group. So, for example, I can replace an aromatic amino acid with another aromatic amino acid. The change will be subtle. There will be some change, but it will not be drastic if I change an aromatic to a sulfur containing or a basic amino acid. So there are a lot of nomenclature that is used among all these 20 amino acids. There have been some names which are really interesting, and there is a little bit of history behind how they named it. And even the single letter code has its own history. So they started with A, B, then they went down till the alphabet, and they've got all the names and the single letter code. Single letter code was devised by IUPAC, and so there was a lot of discussion as to, for example, why tryptophan should be labeled with W when in fact the word tryptophan doesn't even have W, because the word W or the letter W has two V's, and those two V's look as if like two rings approximated view, and tryptophan is an amino acid that's made up of two aromatic rings. So this looks like W. So they labeled it as the single letter code W. Some of them are phonetic. So one of the interesting thing is glycine. Glycine is the smallest amino acid, and one of the plant that has the highest source of glycine is called glycine max, which is our humble soybean. So there are a lot of interesting tidbits and factoids that you can look up based on the names and based on the single letter and the three-letter amino acid code. It's a mnemonic. You can remember these names in some way or the other. And certain amino acids are nothing but the amide form of the acid or another amino acid. Like, for example, aspartic acid, the amine form is asparagine. Glutamic acid, the amine form is glutamine, so on and so forth. So when it comes to the biophysics of proteins, you have to remember that when two amino acids come together, they form a peptide bond. This peptide bond is extremely stable bond. It's much more stronger than a double bond, and it releases one water molecule because this hydrogen and this OH group, they are removed to make the peptide bond. And this peptide bond made up of four atoms, O, C, N, and H. These four atoms, it's a planar in structure, and this planarity of the peptide bond determines what type of protein secondary structure that particular these two amino acids can take part into. So when you are looking at a polypeptide, what you will see instantly in the primary sequence is that the side chain R1, R2, R3, R4, not all of them face on the same side. It's staggered one after the other, and that is how the stereochemistry works to fit in the entire polypeptide chain in a primary structure. As I said, this peptide bond is pretty strong. You can see this gray shaded area, which looks like a rectangle. And that is because this particular bond, this particular area is pretty planar, which means you cannot have a rotation or this bond cannot be rotated. So all these biochemical and biophysical methods of bond length, bond angle determination is extremely important to understand what type of conformational change a protein will undergo when you are trying to inhibit with a drug molecule. So you can see that the bond length is 1.33, which is a little bit different from a single bond or a double bond. So this is how the bonding information is shown, and it is having a partial double bond character, and because of which it is pretty rigid in nature, and it takes a huge effort, or it's an energy intensive process to break a peptide bond. Now, the peptide bonds, when you're looking at it, even though it is still planar, you can have either a trans configuration or a cis configuration. Both are technically possible, but in proteins, we see more often a trans configuration, because in a cis configuration, you will see automatically that the R groups will clash with each other, especially if it is non-glycines. If it is glycines, then the R group is simply a hydrogen. And so there is a lot of space for two glycines to be placed one next to another in a cis configuration. So if you imagine the entire polypeptide in a trans configuration, the magenta or the purple spheres as the R group, and this is the peptide bond shown in the rectangles, you will see that there are these black arrows. It is only these bonds that can be rotated. And these bonds, rotational bonds, we call them as torsional bonds. So torsional bonds are not made up of four atoms or four molecules, but basically they are made, it's an angle made between two planes. So ABC is a plane, BCD is another plane. So when we look at the parameters, and when you look at the angle that's made by in a torsion or a dihedral, we call it in proteins dihedral bonds, you have a certain degree and you have the signage which indicates in which degree it is rotating or which particular way it can be flexible. So the rotational bonds are phi, psi, and omega, and the phi and psi are the most important rotational bonds using which we can determine whether this particular polypeptide chain can form a helix or a beta strand or something else. So if you look at what type of bonding leads to a psi or phi or omega, you can see with the psi, it's the CN C alpha C. These four atoms together, they form a torsional angle. N C alpha CN forms the phi, and C alpha CN C alpha. So the bonds between two C alpha atoms is the omega angle. So the C alpha, what do we refer to the C alpha? I'm going to go back to the amino acid, and the nomenclature is that this particular carbon that is having a valency of 4, bound with the R group, bound with the carboxyl group, bound with the amine group. This carbon is called the alpha carbon, so C alpha. This carbon is the C beta, and then this is the N alpha. So like that, every atom has a particular nomenclature: alpha, beta, gamma, delta, omega, epsilon, and so on, the Greek letters. So what you have to remember is this C alpha is the most important carbon to which the R group or the side chain is attached. We call all these that are shown in blue as side chains. Okay. Now, this particular person, you might know. Can someone know what his name is? It's probably already there on the slide.
G.N. Ramachandran.
G.N.
G.N.
Ramachandran.
Ramanuchandran,
Ramachandran.
Indian Institute
Yeah.
of Science, Bangalore.
Well, basically, he was in University of Madras, crystallography department, and then later on he went to Indian Institute of Science. So he was the youngest faculty recruited in Madras University, which is shown here. And when he came back after his postdoc, he ended up publishing in 1963 the stereochemistry of polypeptide chain configurations with using only the collagen structure as the basis. Now, at that point of time, he uh, came up with the structure for the alpha helix, and he used that particular structure and determined what all permissible dihedral angles can be. That is for the phi and psi, because these are only two bonds that can be rotatable. What you have to understand in the biophysical aspect is that when you have an entity in a three-dimensional space, and then you have another entity which is bound to this first entity, and then you have a third entity bound to the second one. As and when you keep adding more, the steric hindrance and the free degrees of freedom for the first entity, it's lost, or to a large extent it becomes very rigid. So when taken each amino acid individually, they have a lot more flexibility, but when they start making polypeptide, or when there's a chain of amino acids, you have very less amount of maneuver range and moving range that it can do. So G.N. Ramchandra, or GNR, understood this, and along with his two students, Ramakrishnan and Sashi Sekharan, they ended up coming up with something called as the Ramchandran plot. So this Ramchandran plot tells us what are the regions that are permissible for this psi and phi. And intuitively, these sci-fi angles are very specific to certain secondary structures like antipal beta sheets, parallel beta sheets, alpha helix, or left-handed alpha helix, right-handed alpha helix, and so on. And what the one that you studied, collagen triple helix, is here. Now there are three regions, as many of you might know from your basic biochemistry: the allowed region, generously allowed region, and the disallowed region. Any amino acid that has a psi angle in this means there is an anomaly that has to be rectified in the protein structure when they determine it using X-ray or NMR or any method, experimental method. So this was, as I said, when very few protein structures were available. In 1958, we had the first crystallographic structure of myoglobin. This was with a resolution of 3.5 angstroms, or like more than that. So what they could figure out was this long tube-like protrusions, which almost look like something that you would see on the street when you go for a walk in the morning, on the roadside, on the sides. And this particular model, the one that figure that you see the photo of it in the middle, was made with clay. All these are clay models, and Max Perutz and John Kendrew, what they did was they had this model kept in the MRC laboratory in UK, and a couple of years ago, British Museum bought this model and they displayed it, saying this was the first breakthrough in protein structures, and because of which we have advanced a lot with, especially with identifying drugs and lot many things. So this was pretty paradigm shifting work. Now, for those who were born in the recent times and who have studied biochemistry, it would be very easy to understand that, okay, this was a structure, they had a crystallographic method, they identified the structure, and then they solved it. In the 1940s and 1950s, at the time, World War II had just got over, and physicists, they were disillusioned after the atomic bomb, and they were looking for what other things in natural science, which is what biology also comes under, was something that they could read more, understand more about. And it so happened at that point of time, because none of the structures were available. 1953 was when we have the DNA structure, 1958, the first myoglobin structure. So in the 1940s, people thought that biology does not follow any of the physics laws. Biology does not follow any of the chemical laws. So the reason I'm saying this was the most paradigm shifting work was because suddenly they could uh, see that a highly non-symmetrical object, you don't have any symmetry within this molecule, a non-symmetrical object, they can form an ordered structure like a crystal, like a salt or a sugar crystal, and you would be able to see all the different things in the three-dimensional structure. And it so happens that all of them follow the same laws of physics and chemistry. So suddenly it was like an amazing thing happened in 1950s, and people started realizing that by 60s that you can use this method to do a lot more things about protein structure. So suddenly what came to light was that proteins are not simple molecules. It's highly complex, and they can form symmetrical ordered crystals, which goes against the logic that they had earlier. So many of these in that particular period were paradigm shifting work. As we saw earlier, there are four levels of protein structures: primary, secondary, tertiary, quaternary. We'll see somewhat about protein folds between this secondary, tertiary, and quaternary, or between tertiary and quaternary structures. So I'm not going to go into detail. This is a primary sequence. This is the primary structure. So this is the insulin, the sequence, and it shows what type of disulfide bonds are made between the residues, between the cysteine residues, which is the starting residue, which is the end residue. This is all the information that's there in the primary sequence. It looks like a multicolored bead. So Frederick Sanger, he identified or determined the sequence of bovine insulin, which he got the Nobel Prize, and he determined the Sanger sequencing method. And he is the only other Nobel laureate who has who got another Nobel Prize for identifying the Sanger sequencing method for sequencing the DNA. So he was able to identify what all residues and what all disulfide bonds the cysteines make. That is the primary structure. Now, as we saw from the biophysical aspect, biochemical aspect, amino acids have a characteristic amine group and an acid group. And because of this, certain amino acids have some physicochemical properties of being hydrophilic, hydrophobic. So amino acids that are hydrophobic, they will try to find other hydrophobic amino acids and try to expel water between them. And what happens is when you have a primary sequence, it will collapse and it will start forming a globular structure, or try to form a structure where you have a hydrophobic core where there are no water molecules, and on the surface you have a hydrophilic, all the residues that are hydrophilic in nature. Why is this important? Because in the cytoplasm inside a cell, it's basically aqueous. It's a very jelly-like. It's filled with water, but not everything is water. But then you need to expel water out from inside the protein structure to form a stable conformation that can have a function associated with it. So when the main chain folds into the interior and the side chains are protruded out, that's when the magic happens to bring the function in the three-dimensional structure. So the most easiest way to bring hydrophobic residues inside and bring out the amino acids that are hydrophilic to the outside are by using the conformation of a helix or beta sheets. So a helix, and this is the paper that talks about how Linus Pauling, even before the alpha helical structure was identified, he gave a model of alpha helical structure and beta sheet. So they called it pleated segments and helical segments in a secondary structure or in a protein. So they are one level up from the primary structure, and it just so happens that the helical structure is the most stable secondary structure among the two, that is a beta sheet and alpha helix. The reason is that when you are having a twist or a twisted structure of these helices, as you can see all these white, the main chain part or the interior of the protein is all hydrophobic. And the ones that are magenta here, these are the side chains that are protruding outside the helix. So what happens is these water molecules will interact with these, but not with any of these. This will prevent the water molecule to interact, and thereby it makes a very stable structure. Now, alpha helix comes in three different flavors: a 310 helix, alpha helix, and a pi helix. Depends on what is the bonding information or how they are forming. Most common one is the alpha helix, but then you do have 310 helix and pi helix seen in many proteins. You can automatically understand. I make a very crude analogy that if you take an alpha helix and you stretch it out, you get a 310 helix. If you compress it, you get a pi helix. But then that's not the only way, but there are many other quantitative ways. I'm not going to go into detail. Now, because let me go back here. Now, because there is a hydrogen bond being formed, and hydrogen bonds in basic chemistry has a directionality. The hydrogen bond itself is a mini dipole. You have a negatively charged oxygen making a bond with a positively charged hydrogen. You When you take a helix, it automatically becomes a huge dipole. So it's called a helix dipole. All these things you might know from your biochemistry again. And we always see right-handed helix rather than a left-handed helix. And we have different types of helical patterns um, in nature. And we have different ways in which what is the rise and the pitch in the helix. All these are pretty standard measurements that you can do for a helical structure. Why are these measurements important? Because let us say during drug design, you want to modify a particular protein by protein engineering methods, and you want to change something. You want to make sure that the secondary structures are not disturbed. And in that way, the parameters that determine the alpha helix, the rise, pitch, the distance, number of bonds, they are preserved. You can change the amino acid, but if it's preserved, then the protein function is preserved. So helical structures are the most, as I said, are the most common, and they come up with different flavors. You have the alpha helix, which is very common. The pitch is 5.4 nanometer, whereas 310 and pi helix are rare, but then you do see examples of it in many different proteins. So because of the physicochemical property of the protein, which is made up of the amino acids, you have certain amino acids that are good helix formers, certain amino acids that are poor helix formers, and then using that, a very crude way of understanding came up using something called as a helical wheel. So all you see is a helical wheel. We know that the distance or every amino acid is placed at least 3.6 or 36 degrees to each other. So you can actually complete a circle with the 360 degrees. And then you can find out, like for example, this is completely hydrophilic. This one, there is an imaginary axis. As you can see the laser pointer. This part is hydrophobic, this part is hydrophilic. Whereas here you have majorly hydrophobic amino acids. So these are all, oh, sorry, I'm saying the other way around. This is hydrophobic, this is partially hydrophobic and hydrophilic. This one is completely hydrophilic because you have more positively charged residues all over the helix. So we are looking down. Wherever I place the pointer is where I'm looking down the helix and I'm seeing how the helix is rotated, right? So you have different types of helices, and all of them either are present in the right-handed alpha helix here or the left-handed alpha helix here in the Ramachandran plot. And the collagen triple helix occurs here. Now, the next secondary structure, as you might have seen, is in other lectures, is a beta strand. This is a beta strand. This is a beta sheet. So this distinction is mostly lost when we talk about secondary structures. Unlike a helix, beta strand is indicated with the arrow, and this arrow indicates the directionality and of the sequence itself. So this is the first sequence, second sequence, third sequence, fourth sequence, and so on. So the arrow indicates how the sequence is moving. Now, immediately we'll see in this beta strand that it goes up, down through this way. And if you are looking at, instead of this, if I move it this, you'll automatically see something like hills and valleys. Not a huge hill and not a huge depth valley, but something like that. And that is what is called as a pleat. So the most easiest analogy that I can think of is you might have all seen the women in your house, your mom, sisters, everyone wearing saris, and the pleats are nothing but it is kind of like a folded structure. That's why it's called beta pleats or beta sheeted pleats, and those names you will see in literature. Now, because there is a directionality involved, unlike a helix, you can have two types of hydrogen bonding in a beta sheet, either a parallel uh, uh, beta sheet or an anti-parallel beta sheet. How do we know this? Is only by looking at the hydrogen bonding information. In the anti-parallel beta sheet, you will have sequences going, one going down, the other going up, or vice versa. And the hydrogen bonds are perpendicular to the beta strands. Whereas in parallel beta sheet, they will be staggered. The hydrogen bonds will be staggered. Okay, so this is a side view where you can see the pleats, and parallel and anti-parallel beta sheets are extremely common in many structures. So when you have a beta sheet, imagine the beta sheet, the end-to-end folding onto itself, and that will make it a barrel. So what you see here is a beta barrel. Here you have beta sandwich. So this beta is sandwiched between two domains, between two helices and this another beta rich domain, and that's called as a immunoglobulin domain. So suddenly now we come to the area where if more than one beta strand is present or more than one helix is present, we are giving a specific name to it, and those are where we come up with the topic, the protein fold. So all those regions that are not a helix, not a sheet, they are loops and turns. And what you see here is a topology diagram. A topology diagram helps us to identify what type of secondary structures are there, what is the starting and what is the end of the secondary structure, and what type of architecture it can take part in. So what you see here, the numbers is the third residue to the 22nd residue forming the first helix, 48th residue to 54th residue forming the third beta strand. So since all these six beta strands are placed one next to each other, and you can see they are anti-parallel. Why? This is going left to right, this is going right to left, left to right, right to left, left to right, right to left. If all the arrows were pointed in the same direction, that's a parallel beta sheet. So these beta sheets are sandwiched between two helices. Okay. So you have these loop regions where depending on how many residues are there, you have different types of loops, you have different types of turns, and there is a huge amount of function that is present in the loops. And that is why the loop regions are the most important part in a protein structure. When you're doing drug design and when you're trying to identify where the inhibitor will bind, these loop regions are more mobile compared to the alpha helix or the beta beta sheets. And it is these loop regions that will kind of flop around, they'll move around depending on how many residues are there between this beta strand 1 and beta strand 2. You have like two residues here, or you have three residues. It depends on what is the length, and more lengthier the loop is, the more floppy it is. There is also other types of structures that we see in proteins. There are beta bends or reverse turns. There are different types. All these type 1, type 2, type 3, type 4. All these names are given by IUPAC to identify which one has what type of structure. Once you have the secondary structures, we come to the area called as motifs. So motifs in like literal meaning is patterns that are repeated multiple times. So for example, I can't see any of your videos, but I'm pretty sure all all of you are wearing something that has a motif in it. Like for example, I'm wearing a pattern that is repeating multiple times, the checked pattern or the plaid motif. This is called a plaid shirt. Similarly, in your the ladies or the woman, you can see in your dress, there will be some motifs that are repeated multiple times or patterns that are repeated multiple times. This is called a motif. Now, in protein structure, a motif is not a repeated pattern, but it is a super secondary structure. So what you see here is a very famous motif, an often repeated motif that you see in many proteins, a helix, turn, and helix. So this helix, turn, helix is a motif where the position is present like this. You have a helix, my forefinger is a helix, then my crevice here is the turn, and then my thumb is the other helix. So this particular helix, turn helix can squeeze, can open, and it can bind to something. Most of these helix, turn helix motifs bind to DNA because they can go and hold on to the DNA. And they have a particular function associated with it. So there are other motifs also like beta, alpha, beta. So this is beta, then alpha, then beta. This is a motif. Now, why do we call this as motif? Because they are supersecondary structures and this motif can repeat in some other place as well. Okay. So you have a Greek key motif and we'll see what the Greek key is. So this helix turn helix is very important motif in the protein called calmodulin, where the calcium ion binds to this particular region. And automatically you will see that this region is a loop. It is not in the helix. Excuse me. So this helix turn helix, as I said, the loop regions are very floppy. That is where the calcium is making an interaction. And this is the atomic level interaction of the calcium, making all sorts of bonds with all sorts of residues and even a water molecule. So the calcium binding motif, the helix turn helix motif, they are present in many, many different proteins and they are majorly all helical motifs. And the most common example is calmodulin, which is binding to a calcium. Then there are motifs made by beta strands. This is a sheet motif. So you have a hairpin beta motif, a Greek key motif, or beta alpha beta motif. So you have alternate alpha beta strands in antiparallel orientation that make a motif. So again, that is a hairpin beta motif, then beta alpha beta motif. Now, depending on where the helix is, if both the beta strands, you imagine them on the same plane on the sheet of paper, either the helix can be above the paper or the plane or below the plane. So both can be technically present, but we see only one most often in nature. Now, what you see here, this is the Greek vase, and you would see that there is this particular line at the rim of this vase that goes like this. This particular motif or the pattern in the pottery of Greek, ancient Greek, is called Greek key motif. And we see that same pattern in the protein structures. So you can see that it goes like this. You see that this beta strand, if this is beta strand 1, beta strand 2 is sandwiched by beta strand 3 and 4. So this is a Greek key motif. And Greek key motifs have a huge function present in it. For example, in the protein crystalline, which is present in our islands, they are majorly made up of Greek key motifs. So this is how the Greek key motif looks like, and this is exactly the same conformation that you see on the proteins. Now, these naming conventions for these motifs are something that we have given because we are looking at it and then seeing similarity or something relatedness with something that we know from our daily life or some other context. So that's the reason why you have Greek key motif. There was no Greek involved in this, or there is no key in this motif. This particular pattern in textile, in pottery, in architecture is called Greek key. That's all. So now multiple motifs can come together and can form something called as a domain. Not always, but This is the next level up. And there is a reason why we are talking about domains is because when we are looking at a protein fold and we want to identify what type of fold it will take part in, then or what type of function that fold will have, you want to know whether there are domains in the fold or whether the domain uh, present with many different motifs. So, for example, this particular structure that you see is made up of beta, alpha, beta, alpha, beta, alpha, beta, alternate uh, beta, alpha, beta motifs. But together, all of them form a single domain. So the motif alone, like in the calmodulin protein, the motif might be the region where the protein function happens, but the motif alone cannot be a structure enough to carry out the function. It needs other supporting things. You can think of this an analogy like you have a house, you are in your house. The most important part in a house to call a building as a house is the kitchen and the bedroom. Rest everything else, if you have, if you don't have, that's okay. So if you think you for a house, the important part is the kitchen, and you just build the kitchen, that you don't call it a house. We don't call it a home. You need to have a living room, you need to have the closets, you need to have as another living space or something else. All together, you call it as a home or a house. Same logic, the motif alone can have a function present in it, but the motif alone cannot function unless there are other supporting elements that are there in the structure. So similarly, if the function for this particular protein is present in the loop region, it needs the support of all of this to have a stable structure with which it can function like that. So domains are nothing but independent units within the protein where a function can be assigned. And these are independent because you can have single domain proteins, you can have multi-domain proteins. So like for example, EGF domain. Just that EGF domain is enough to carry out the function. However, the EGF domain, along with something else, and the serine protease domain is seen in urokinase. Plasminogen consists of multiple things along with the serine proteinase domain. So there are 5 Kringle domains and then a serine protease domain. Factor 9 has a calcium binding domain, 2 EGF domain, and then the serine protease domain. So depending on the protein, depending on the function, you have multi-domain proteins or a single domain protein. So multimeric proteins are made up of the quaternary structure, which is like uh, from secondary structure, they fold on themselves. Their architecture is kind of dictating how the structure will be, and then it ends up binding to something else. It can be a, like in this case, hemoglobin is a heterodimer, meaning the cyan and the green are the same copies of the B chain, and together they form a heterodimer. It might be the case in like in glycerol 3-phosphate dehydrogenase or in GROEL complex. It is a copies of the same protein repeated multiple. So it's a homooligomer or a heterooligomer. So it cannot function as a single chain. It has to function as a 4 chain. Then you call them as multimeric proteins. Now, why is this information important? Because when you do drug design, you might have the structure of a single chain in PDB or the main protein chain, primary, secondary, tertiary structure available in some literature. But if that particular functional stoichiometry, which is what we call in biophysics, the stoichiometry, whether it functions as a monomer, functions as a dimer, trimer, hexamer, or multimer. Until unless we know that, you whatever drug design you're doing is not of great use. For example, grow EL you take. It has 1, 2, 3, 4, 5, 6, 7 chains of the same protein. So it's a septamer. Now let us say you only have the monomer and you did not know that it binds to 6 other copies or 7 other copies of itself and makes a full protein. It is this complex is functional. Let us say you didn't know about it. You might think that this region is the region where it is very important for the protein to do its function. But in reality, that particular region is unavailable for anything to bind because the neighboring chain is binding to it. So looking at the stoichiometry, we can figure out which particular region of a protein is interesting, which particular region is against which you have to design inhibitors or go ahead and design a drug. So to understand this quaternary structure and to understand how many domains are there in a structure, we come up with a flattened representation of a protein structure called as a topology. So topology is a two-dimensional abstraction. We are just seeing topology, what it is. Like for example, you have a 4 beta strand and with a short helix. This is the topology of this protein. So what has happened is you have kind of took that protein, you have stretched it out, laid it on a sheet of paper, which is a two-dimensional representation, and you can see the connections of how it binds. Based on that, you can actually say what type of fold it is most probably going to be and which residues are important. So topology is not the same as just sequentially arranging the secondary structures. Like for example, here the first beta strand is connected to the second, but first is binding, making hydrogen bonds to the third. And that is why this is the topology rather than putting 1, 2, 3, 4, 5. That is not topology. Similarly, the Greek key motif, you can see the Greek key motif here, you can see the Greek key motif here. And again, how these bonding information is presented. And immediately it will be clear if you see this and you say, oh, I don't see a Greek key motif here. When you lay it out in a topology diagram, you will see a Greek key motif. There's a question, do multimer proteins perform single function or multiple functions? Usually, every protein ends up working for one particular function. So even if it is a multimeric protein, it will be doing only one function. There is a difference when I say one function and a difference when I say that it can bind to 10 different substrates or 10 substrates. Because in a decamer, let's say, a protein that has 10 chains, you will have 10 places in the chain, in the each chain where the activity can happen. So in that case, the effectiveness of the protein has increased tenfold, but it still is doing the same function for all the 10 chains. Okay. So like here, for this particular protein, you have the secondary structure label, the color label telling us which is the domain, which particular part is involved in what type of hydrogen bonding. This is for dihydrofolate reductase, a very popular protein in E. coli, and you can find out whether there is any disruption in the protein structures. Now comes to the next part where based on the protein secondary structure, the domain, you can have different classes of proteins. So proteins that are mainly made up of alpha helices are called all alpha proteins. What you see here is a coiled coil, a 4-helix bundle, or a ribonuclease inhibitor, a horseshoe shape, one which has lot many helices, a huge molecule. So you have only made up of helices, and then the helices are connected by turns or loops. So all these images are from the Brandon and Too's book, and it makes reading very enjoyable. And let me tell you that Brandon and Too's is a prescribed undergraduate textbook for biology students in many different universities. So it's a very easy to read book, and they have written it like a story. So just like reading a novel, you can read the entire book very easily. Coiled coil, when this is a coiled coil. So these two proteins are making an interaction. And if this, let's say they twist and you are having a twisted long coil, you will get something like this. And if you have three helices that are twisted like that, then you get a triple helix, and that is the structure of collagen. Now, coils, as you can see, that they form long stretches, and that is why our hair, our nail, our skin, those proteins, they are mainly rich in coil alpha helices because they give structural stability to the protein or to the entire cell. So we have lot many different longer coil regions for hair and for skin, but then you have some transcription factors where shorter coil helices can be seen. So in many cases, there is a 7 residue repeat, which you can predict, and they are most probably going to be a coil because in the 7 residue repeat, all the residues that are on the inside of the coil are hydrophobic. So all those that are on the outside on the out either ends are hydrophilic in nature. So there are different types of models, knobs in holes or ridges, a model in which how these coils are arranged one up one next to each other. So there is some kind of angle that's made. So the 4 helix bundle is the coil, but made up of 4 helices. So all these green spheres are nothing but hydrophobic residues which are making an interaction to bring the 4 helices together, and the hydrophilic residues are the ones in red that are making a bond to the water molecules. So the hydrophobic core makes sure that this 4 helix bundle remains stable. It so happens that in drug design and in protein engineering, the most easiest structure that you can create from scratch is a 4 helix bundle because these 4 helices, 6 helices are very, very stable structures, and helix by itself is a stable structure. And so you can create new proteins from a helix, starting with a helix. So this is again, you can have, depending on anti-parallel orientation or parallel orientation of the helices, you get different types of architecture. Now I'm using the word architecture here because this is the right time to introduce that these these are no longer motifs, these are no longer domains, but then there is some kind of a pattern that is repeated in many different proteins for these kind of layout or topology, and that is why we call it as an architecture. Skip that. Okay, now coming to fold. What is a fold? Fold is nothing but the arrangement, the three-dimensional arrangement of the secondary structures for a protein that can carry out its function. If a protein does not fold in the required way, then the protein function cannot happen. The protein is non-functional, and there are mechanisms inside the cell that will convert the unfolded protein to a folded protein. And those proteins are called as chaperones. So the most easiest one or the most popular one is a globin fold. Globin fold, as the name suggests, it's an all alpha protein. What you see is all helices. And this globin, you prefix it myo or hemo, all those have globin folds. So the globin fold can bind to a particular inorganic compound, and it makes the arrangement or the architecture of these helices makes what is called as a globin fold. So the globin fold is made up of 8 helices labeled here A, B, C, D, E, F, G, H, connected by short loops, and in which the myoglobin and hemoglobin at this particular point binds to a heme group. Okay. So in the beta domain structures, you have structures that are all beta. Either it can be all anti-parallel beta or all parallel beta, but these form a barrel. As I said, if you take the, let us say you have 8 beta strands, the first and the eighth, if they are coming closer to each other, it just becomes a cylindrical structure, like how you see here, and they call it as a barrel-like structure. Now, what you can see is you can have many different types of barrels because unlike 4 helices, where the connectivity is not diverse. Here in up and down barrel, you can have many different kind of connectivity between the beta strands. And you can have connectivity like this, a Greek key barrel. So you have two Greek keys making a barrel, or it can be a barrel with like this type of simple configuration, and you have an empty open space. Automatically you will ask, okay, where are these barrels present? Now these barrels, because it is cylindrical, it is like a tube, you can immediately imagine that, let us say it's a tube and you are making it pass through something, then it becomes like a pipe that connects the outside world to the inside world. Now, that particular analogy is what exactly happens in a membrane protein. So, membrane proteins are embedded in a membrane. And let us say this vertical region, you have a helix and you have a lipid layer, and here also you have a lipid layer. So this is the extracellular part and this is the intracellular part. Now things can go between back and forth in these barrels, and that makes it very interesting because these proteins are now involved in a different type of function called as signaling, and the up and down barrel or Greek key barrel makes the protein function in a different way, and it will allow only certain types of molecule to pass through depending on the size. So you can have large barrels, you can have very small barrels, you can have different types of barrels. Again, these are all different types of architecture that can be seen. So in gamma crystalline, two Greek key motifs form. This is one Greek key, this is another Greek key. So two Greek key motifs form a domain, and two such domains form a functional unit. So this is a two-domain protein with four Greek key motifs, and each domain has two Greek key motifs. So this is an independent unit, this is an independent unit, and these two are connected by only one loop or one region. Then, if yeah, almost all of you might have eaten a Swiss roll. So those Swiss rolls, so they're called as jelly rolls. So jelly rolls is nothing but you have this type of an arrangement where you have alternate beta strand, the bonding between them, 1 and 8, 7 and 2, 6 and 3, and 5 and 4, and they roll over each other. So one is inside the other. So that's called a jelly roll barrels. So those were two types of folds. One was all alpha fold, and other was all beta. Now comes a mix, alpha slash beta and alpha plus beta. So this is a protein. What you see here is a alpha beta barrel. So this barrel part is made up by alpha helices and beta strands, and uh, sorry about that. And but from the color, you can see that it is a three domain protein. So this is green is a domain, red is a domain, blue is a domain, and it is a multi-domain protein. That is something on the chat box. How synthetic peptides ligands help in binding to motifs, alpha helices, beta sheets, or any limitations. Synthetic peptides, again, it follows the same physical and chemical laws that we follow in the L amino acids and the natural proteins. So the only difference is how novel the sequence is. If it is a synthetic peptide, which the sequence is not seen in nature, then you have a completely new peptide that can bind to something. And one of the AI tool that comes up does a job of finding out new sequences that can have the same structure or something similar is RF diffusion. Or and then there is one more EVO bind, which can give peptides that can bind to a particular protein. Okay. The limitation in that particular case is that there is a lot of false positives. The success rate is like 30%. So 70% of the structures will not bind at all. So that is one downside of the synthetic peptides. And most of the synthetic peptides, they end up being precipitating inside the cell. They form inclusion bodies. They cannot be solubilized. There are a lot of issues with synthetic peptides. So alpha slash beta is nothing but the protein fold that has both the alpha helices and the beta strands, but they are interlinked with each other. For example, here we have a beta alpha, beta alpha, beta alpha. So if you look at the sequence, there are beta, then followed by alpha, beta, alpha, beta. So this is called alpha slash beta. Now I'm going to go back here and I'm going to show this is a alpha slash beta domain because it will have alternate beta, alpha, beta, alpha, beta, alpha. But this domain is all beta domain because you have only beta strands in this particular domain. Here you have alpha slash beta again. Now imagine instead of this particular orientation where alpha and beta are interlinked, you have one region in the domain that is completely alpha helical, like let us say here, and completely another domain that is completely beta in nature, but it's a single protein. That is called a alpha plus beta structure. So there are many different other complex motifs. Now, this is the one motif that is very important for drug design. Why? So look at this. There is a beta strand, then alpha, beta, and then the alpha is connected to a beta, but then the orientation is completely different. It's coming somewhere in the middle of the alpha and the beta. What this creates is an opening or a crevice. So, okay, that's what it's for me. So you have two motifs, and in between them you have a crevice. Now this crevice, like for example, in this particular place, all these crevices, wherever you see this arrow, are the functional region of that domain. So this particular protein or any protein, when we look at the arrowhead, so this came up with a general rule of thumb that if you have a alpha slash beta proteins, a fold that has a mix of alpha and beta, then look up at the loops that are at the end of the beta strand. So beta strand starts at this point and ends here at the arrowhead. So wherever the loops are there at the arrowhead, those regions are the active site regions of a protein. And if, let us say, during drug design, you come up with a fold or you're looking at a fold and you don't know anything about the active site, and if, let us say, you find such type of crevices, then it's most likely that those crevices are where the active site region is going to be. So from sequence, we went, you have the structure, and the structure, you have the function, where you have a region where is the most important part which can bind to something like a drug. So this particular sequence dictates structure, structure dictates function, is the dogma in protein structure. Okay. So how are we doing? Do you have any questions? Any queries? Okay, so no session is going great. Okay. We'll finish the lecture in another 15 minutes and then we'll have a demo. Okay. So protein, as I said, they fold. They form many different folds. There are about, as I said, the protein universe is about 1 trillion in size, but the number of folds is about 1,000 or maximum 1,500. That's it. So then the question comes, why so many less number of folds? Why we don't have 1 trillion folds, every protein folding into a different architecture? There are two reasons for this. One, that we have mapped only so much about the protein universe that we know only so many folds. In crystallography, protein structure field started, and in the 50s and by the 80s, the crystallography field advanced. They could speed up the crystallography process. NMR came up where most advanced methods came up. And the speed at which a protein structure could be determined ended up accelerating a lot. And because of that, what happened was new folds were being identified. And now, if you are going to solve a protein structure, it's 90% or 99%, it would already belong to a fold that we already know about among the 1,500. So we have the first reason is that we have only so many folds that is allowed in nature, and invariably a protein sequence folds into one of these 1,500 and not protein folds, like the globin fold or Timbaral fold and so on. Okay. The other reason is that protein universe is so vast that we, just like our galaxy and astronomy, we have seen only very tiny bits of the universe. We have yet to see a lot more things about the universe that we don't know if there are other folds available or not. And so with time, with more methods being identified that can speed up the process, especially computational process with AlphaFold and all this, the AI methods, we think that we can probably identify new folds. But whether we'll really find a new fold, that's a big question. Now, this is a cartoon that shows an acrobat or a contortionist who is twisting himself, and within a split second, he folds into a very stable structure. And although this sounds very comical, but that's actually what happens in reality, that a primary structure forms a tertiary structure within like a matter of seconds. Okay. Unless it's like 1,000, 2,000 amino acid long sequence, those proteins take maybe like minutes to fold. So what we know now is in the protein folding is that on the y-axis is the energy. Here you have very low energy. Here you have very high energy. You have these valleys and hills. Okay, so uh, I'm pretty sure most of you have been to Himalayas and have gone to one of those hill stations, either in the south or in the north or in the west. Anywhere you have mountains, that's the hill station. What do you see on the hill station? You have mountains, and in between mountains, you have valleys. This is exactly what you see there. So if you take a slice of the geography, you have mountains, you have valleys. So imagine you are at the top of a mountain, that is a very high energy state. Proteins don't like, or biological molecules don't like to be in a high energy state. This is what I'm going to say next is related to what I'll be showing you tomorrow. But just like our human body, we have many different orientations in which we can fold ourselves, like a contortionist in a circus. And even though there may be like hundreds of different ways in which a body can fold in anatomy, very different, bizarre. So please believe it or not, you will see lot many different unusual anatomies of human. But there are two conformations where the human body is the most comfortable and spends very less amount of energy. Can someone tell me what are the two orientations or two conformations of the human anatomy in which we are very, very comfortable? One is right now what we are doing and what we call it as. What are we like doing, all of us?
Is a sitting position.
We are all sitting. We are very comfortable. Okay. In a movie theater, in front of the TV, we are sitting. What is the other conformation in which we are very, very comfortable?
Straight lying in bed.
Yes, sleeping, right? So these two conformations are very, very comfortable, not just physically, but energetically as well. So proteins don't like all this contortion structure. They are high energy. It will try to unfold and it will go to a state where it is highly folded. It's very happy, energetically very stable. So it needs to be in this particular place. So if you look at the protein landscape, how does a protein go from unfolded, which is the primary structure, to a tertiary structure in a matter of seconds or not even 1 second, it's like millisecond, sometimes it's like microsecond, and without anything telling it to go into that structure. So this is where the physics part comes in, that the rules that are there of how a protein can fold into a particular fold, like a globin fold or another fold, is already embedded in the structure. And so if you change something in it, the structure will change. So again, the dogma, sequence dictates structure and structure dictates function. So if you change something in the sequence and the structure changes, then the function changes, right? So this unfolded is inactive, whereas this folded is active. This has the function. So there are a lot of many different theories that have been used. There are a lot of different popular ones that have enabled us to understand how a protein folds and what kind of mechanism it is. Now, here's a paradox. Okay, again, this is the same thing, not the paradox, but what happens is here you have very high entropy, you have very low entropy here, and in between the protein, when it goes from the unfolded to the folded, it goes through a stage called as a molten globule state. Okay, so those intermediate states are okayish, but then they are not favorable. They are very transient structures which we can't capture through any experimental methods. It's only a theoretical way of understanding them. Experimentally, it is very difficult to even identify the molten globule. But you can think of an analogy where you are melting metal. You have molten metal, so you have molten metal here, and after some point of time, the metal cools down, and you have a place where, depending on what is the shape you give, the metal can take that particular shape and remains in that state forever. So this particular process is driven both by entropy and enthalpy, which is where it goes from a high energy state to a low energy state. So the energy landscape theory is that when you are in a time scale where it is in a very unfolded state, you tend to roll downhill towards the valley for getting to the most stable state. But in that particular path of whether you're going from here or here, this is an ideal protein folding landscape. So you have only one native state. However, you have multiple different landscapes, so might be end up in a trapped local minima, and this local minima is the protein is stable but not so stable. It's just waiting for something it to push it out and go to the native state. So in those cases, we call them as a metastable states. So there are a lot of proteins that are in a metastable state. They change the entire conformation, domains get swapped, and a lot more things happen there, and with pretty interesting things that happen. So in protein folding, the problem that was there for which people understood protein folding, I'll come to this slide later, is that you have many different examples in real life where you have the native protein which can either undergo a thermal or mechanical or a chemical denaturation, and you can go from a native state to a denatured state. Okay, and this process in many cases is irreversible. Once a protein has been denatured, it remains there. You cannot fold it back until unless there's a heavy some other kind of mechanism present. So Cyrus Leventhal, he was studying this biological mechanism, and he made a small theoretical calculation. Let us say you have a protein of 100 amino acids that are connected with 99 or 100 bonds. And let us say each one of them, remember the sci-fi angle in the Ramachandran plot, it can take only certain conformations. So let us say among many different conformations, each bond is like taking only about 3 possible configurations. Then you have 3 to the power 100 different conformations. Those are the entire landscape. So to go back to this landscape, you have confirmation 1, confirmation 2, confirmation 3, confirmation 4, 5, 6, 7, 8, like that. The entire landscape is filled with each one is a conformational spot. Okay. Now, you want to find one structure among all these different conformations. Let us say the protein is able to sample 10 to the power of 13 different configurations per second. Okay. Among all this, every second, like this, it can sample 10 power 13. Even then, it would take about 10 power 27 years to sample all the possible conformations and find out which is the conformation that the protein can fold into. This is called a paradox. Why? Because this number of time is much longer than the age of the universe. And we know that an E. coli can divide in 20 minutes approximately, which means all its proteins should be functional within the first 5 minutes for it to function properly as a cell for the next 15 minutes and then divide again into another two other cells. So this is a paradox. In reality, the rate at which a protein folds and the sampling that it takes is not these many, and it is in a millisecond or even microsecond, and the protein conformational space is so huge that the protein knows how it's going to fold. Now I'm going to go to the previous one. So because of this paradox, what Levinthal had found, and everyone knew that there was a paradox, that proteins fold spontaneously. And if you had seen some animations of protein that are coming out of the ribosome, as soon as a few amino acids come out of the ribosome, they start folding. And they will start folding, 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 folding. And within a microsecond, the functional protein will be thrown out and it will start functioning. So in a physiological condition, the primary structure dictates its three-dimensional structure. So it doesn't wait until the entire chain is there. It starts folding. There are some micro events that happen and it starts folding into structure. Now, very interesting work happened in, I'll come back to all this point. Very interesting happened was what Christian Anifson did. Now, Christian Anifson worked with ribonuclease protein. Now, this ribonuclease protein contains certain bonds, okay, like any other protein. Now, what he found was when he took this ribonuclease protein and he denatured it using urea or high temperature, it became a denatured protein. The protein unfolded, it became a linear structure, primary structure. And then after some point of time, when he removed the urea or when he removed the temperature, he found that the protein could fold back and the function was still available. So in the denatured protein, there was no function. The normal protein had a function. But even after denaturation, it renatured and it formed a normal protein. And he found that this particular ribonuclease does that because the disulfide bonds in that dictates how the bonding has to happen. Now I'll go back to this. Now, what is disulfide bond? This is made between two cysteine residues or two cysteine amino acids. And for those who know this, that there are two amino acids that have a sulfur atom. One is cysteine, the other one is methionine. Methionine doesn't form a disulfide bond because that sulfur is not available for bonding at all. It's only cysteine that can form a disulfide bond. Now, disulfide bonds are extremely strong bonds. These bonds cannot be broken very easily. You need to have a reducing agent that can break it down. Now, what Anifson did was he denatured it, and then after reducing it to a denatured molecule, he oxidized it and he started identifying among the four disulfide bonds which disulfide bonds are being formed first, the second, and so on. And then he also did studies where if he did not make a particular disulfide available, what happens to the sequence of disulfide bond formation. So to remember how complex this issue is, because you have 6 cysteines, you have like for example, you have 8 cysteines, you can make 4 disulfide bonds. And then if you already have one disulfide, then the probability of the remaining 6 forming a proper disulfide bond is 1/5, and then remaining 4 is 1/3. And if you take a calculation, you will see that there are like 105 different combinations among these 8 that can form a disulfide bond. Clearly, this particular conformational search space of making a disulfide bond with any two cysteines is not happening, and it's not a random process. So this is where it was like lysozyme has 4 disulfide bonds, so it takes about 105 combinations. And even RNAs that Anifson studied. The more the disulfide bond, you see that the number of combinations is exponentially increasing. So this gave the idea that once you, let us say, you denature or you partially denature, you have a rule that is present that you can make certain disulfide bonds form first, and it doesn't matter which two disulfide bonds are forming it. And as soon as the first two are forming, and immediately the second one, the rate at which the second disulfide bond is formed is much faster. The third one is much more faster. And that means the protein accelerates from the high energy state of the denatured state to the native state in a very fast manner, and it knows which path it has to take in the protein landscape. So these two things, the Anderson's experiment and Levinthal's paradox, actually gave rise to a lot more questions than what people thought would have ended up protein folding. Basically, with Anderson's experiment, you can have many different intermediate structures which actually represent the molten globule. And so there are a lot of thermodynamic properties that you can study if you mutate a lysine, mutate a cysteine, and then find which disulfide bonds are being formed. So there were a lot more studies that were done. So this protein disulfide isomerase was used to identify which disulfide bonds in RNAs are more favorable, which forms first, which forms second, and so on. Okay. So then there were a lot more studies with many different proteins, and slowly people understood certain things about protein folding. One, from the unfolded to the folded state, there are certain things that are necessary. One is that it tries to fill up space. The core of the protein should be tightly packed. The hydrophobic core, once it forms, it actually dictates how the rest of the residues have to be placed. And then the secondary structures can be extremely context dependent. It can be present in hydrophilic part, it can be present in hydrophobic part. And depending on where they are, you can have many different types of properties of the secondary structures. Similarly, two structures which look extremely similar in terms of the architecture, you can have different sequences that can have the same structure, which means the fold is conserved, the architecture is conserved, but the sequence can be different. So the helices and sheets, which are basically the building blocks of the fold in among the 1500 folds that we have, you can select which type of helices can form which type of proteins and how they are fine-tuned in pairing and interaction in many different proteins. So this came up with an interesting experiment where how much protein sequence can be changed that will convert a protein to change a fold completely different. So this was a $1,000 contest, and this was announced about 40 years ago. And there were many teams that competed, and one team ended up converting a protein from a completely different fold to another fold by changing 50% of its residues. So those were the rules. The rule was that you can change only 50% of the residues, but which 50, it's up to the person. Can you refold this protein into something else so that the sequence identity between them is 50%, and you can still get two different structures. So the protein GB1 can be converted to raw protein by changing 50% of its 56 residues. So this was very interesting because now you have a protein that can change fold, and that looks like this Roman or the Greek mythological character called Janus, which is named whose name is what is named after the month January, because Janus is a two-faced god, and he looks at the past and the future, and January being the month that looks at the previous year and the coming year. You have the protein that can have two different conformations with the same sequence. So this ended up starting a lot more protein design methods, protein engineering methods, protein rules about changing a fold or identifying what makes a protein fold into a particular architecture. So I'm not going to go into detail about all this because there are other things that we need to do within the time that's given. But how do you look at all these folds? Well, you can go to SCOP or CATH, and I'm going to go to, let me finish the presentation and then I'll show you the demo. So there is a SCOP, Superfamily, CATH, FSSP. These are the four databases where proteins are classified based on the folds. Each uses a different type of method to classify the proteins. So SCOP is a manually curated method database. So it is a very slow in identifying, or there is a human behind it who classifies it. And there is a automated method, but the human determines whether that is right or wrong. Superfamily uses another type of a method, a hidden Markov model method, to identify fold. Then there is CATH. The CATH, sorry, I didn't explain this. In SCOP, the SCOP stands for Structure Classification of Proteins. CATH stands for Class, Architecture, Topology, and Homology. And you have different types of classes. And then there is a DALI fold classification tool. DALI also has its own database using which you can find out what fold the protein contains. And like, for example, this particular protein is globin-like, okay, and so on. So it will give you what prediction it is. So this particular tool, FIRE, can actually tell you what type of superfamily it belongs to or what type of fold it belongs to. Okay, so the type of proteins, yeah, so this is the CATH database, the website. You have 151 million protein domains that are automatically classified into 6,000 superfamilies. This is version 4.4. So you can search for any protein, like my favorite protein to go is hemoglobin. And if you go to hemoglobin, it will give you the classification 1.10.287.120. So what does this one indicate? One is the first class, which is all alpha. And then you have the architecture 10, then the homology 287, and the topology 120. So every protein gets a 4 code. And then, like, for example, if this is the PDB structure 1AA0, here you can see what type of details you have for that particular domain. You have the superfamily CATH. You can see 1.10.490.10. That is the ID for this particular class and what other proteins are involved. Similarly, SCOP, this is the SCOP2. As I said, since there is a human involved, you only have about 72,000 non-redundant domains representing 861 protein structures. This is way less than 150 million that CATH has. The reason is CATH is completely automated, whereas SCOP, you have a tool called, I forget the name. So it uses the tool to assign the domains, and based on the domains, a human then curates whether it belongs to which particular fold. So you have different class, all alpha, all beta. Since we saw all alpha there, we'll see all beta here. You have propeller, immunoglobulin like beta sandwich. All these names is the next level of classification of folds. Okay, so there are many, many different folds, and into each fold, let's say double-stranded beta helix, you'll have different superfamilies. If you go inside a superfamily, you will have many families, and each family will have many different domains. And a protein can have multi-domain or a single domain, and each domain can belong to a different family and in turn can belong to a different class. Okay, so these two popular tools or databases are very useful when you're looking at a drug target. As we say, as we know, this entire workshop is about drug design. Why are we talking about protein folding? Why are we talking about protein folds? Any drug target that you take, you need to know what class, what architecture, what type of fold it belongs to, how does it fold, and what it is able to have the active site. Where is the active site? Does it have a crevice? Does this particular fold undergo huge conformational change? If it undergoes the huge conformational change, then what is the time scale of the conformational change? Does it happen in milliseconds or microseconds or nanoseconds? And then how big the protein is? Does it interact with some other proteins? Does the fold level information indicate whether it is a multimeric protein or a homodimer protein, or what type of oligomeric stoichiometry does it have? All these questions, the answers will be present in the fold level information. And if you are unable to find a topology for your protein, let us say you don't know, you have a new protein, okay, and that new protein, you need to generate topology. You can go to PDB sum. I'll show you what I'm talking about. Let us say this particular protein, 1KFV. And then if you click on the protein, you will see the topology of that particular protein, the topology diagram. Okay. And based on this topology diagram, you can find out what is the major class, what type of fold it folds as. Okay. So let us say this particular protein is something that is not there in the PDB, and you have to generate topology for it. You can go to PDB sum and you will see this link here called generate. So you have to upload your PDB file and give your email ID, and it will give you a topology for your protein. And in many cases, like for example, let me go back to that example 1KFV, it also gives you reasons like clefts, which we talked as crevices in the structure. So those are most likely active site or regions that the protein binds to a ligand or an inhibitor. If you're doing a drug design, you want to have this thing go and bind to this. It's like a pocket. Okay, so like my AirPods and then the cavity that the headphones can do. So, and then if it is a channel protein, you will see tunnels. If it's a barrel, beta barrel, you will see tunnels. So do you have some space where things can go around, a water molecule to go around, or some ion to pass through? All those details you can find it in PDBsum. There are not just these, there are many other tools that does this, but it's kind of like you have basic information in SCOP, CATH, and PDBsum for to find the fold level information. So at the top page, which is a summary, you will see what type of protein it is. Okay. And if you click on the protein, you can see the CATH classification. It says alpha beta barrel, and then subsequent another CATH classification 110.8.50. Same information will be there in PDB. If you go to 1KFV RCSV, and in PDB's page itself, you can find the fold level information. You don't have to go to PDB sum, but topology will not be present in PDB. You can get it from PDB sum. Okay. So if you click on the structure tab at the top, what we see is the summary page, but there are other tabs in the structure tab. You will see many different information, and in that you will see the fold level information for that particular protein. While it's loading, I can take some questions. Okay, there are 4 questions. What is the significance of reverse protein engineering in predicting protein sequence with reference to unknown protein structure? So when you have an unknown protein structure, the reverse protein engineering is that you want to find out, sorry, it's an annotation tab. So yeah, there you go. So here you have all alpha protein, all beta protein, small protein, and then you have class, fold, superfamily, family, domain, species, and so on. So this is as per SCOP, and then you will have as per CAT as well here. You can see CAT classification, class, architecture, topology, homology. Okay. Okay, so the first question is, what is the significance of reverse protein engineering in predicting protein sequence with reference to unknown protein structure? So when you are having an unknown protein structure, you don't know where the active site is. You have to make a guess. You have to have a confidence as to where the active site is. You can predict to some extent whatever molecules can bind to the protein, but then if you want to have a much better enhanced binding, then you'll have to engineer that particular structure. So that is why the reverse protein engineering ends up playing a role. Second question is, can we classify functions of proteins with reference to that protein structure family? To a large extent, yes. Like for example, all alpha proteins, like if it's a globin protein, then it's most likely going to bind to a heme or some type of inorganic ligand molecule. Beta barrels, they are membrane proteins. So that means they are channel proteins or pores. So with motifs, yes, like for example, if it is the EF motif, which is the helix turn helix, it may be binding to a metal ion. So you can, to some extent, to a large extent, identify functions. How fold-based databases help in identify protein ligand complexes? Fold-based databases don't identify protein ligand complexes. They identify what type of architecture the protein contains. So that in turn can tell you what type of ligands can bind, because that architecture will tell you what is the space that's available for something to bind. Are there any rare folds conserved evolutionary? Could be specific targets, but how to identify? As I said, with many methods that came up, we are not finding that many new folds, but we still have many structures to solve. So there might be some fold that we have not come across. It is still elusive. That's one hypothesis. The other hypothesis is that irrespective of whatever protein sequence you will have a designed protein or a unnatural one or a natural one, you will still be able to fit the protein sequence in one of the folds, which means that there is only so many limited models or architecture that's available in nature, and every one of them, every one of the 1 trillion universe, protein universe sequences folds in one of these. So how fold diversity helps in CAD? I just explained it to the entire talk was on that. So fold diversity tells us if you mutate a protein, if you change something, does it change the structure? If it changes the structure, then how much of the fold is changed? How much of the domain rearrangement happens? What type of interactions will rearrange? And because of that, you need to see if the protein folding is not going into a local minima, which is trapped energetically, not favorable for binding, but you want to get the native state. So getting to know the native state of the fold and the architecture is crucial for drug design. Any other questions, doubts? You can unmute and you can ask. Or you can put it in the chat box.
Yeah, participant. Now you can start your video. You can open your video for a discussion.